Okay? Now, we're going to look at meiosis in some detail. Please be warned that meiosis evolved from mitosis, and because of that, it looks really similar. A lot of the same machinery parts are used, but remember how different the processes are. Now, interphase, cells just living out its life. And that is essentially the same in meiosis as it is in mitosis. We've got, remember, interphase, we got G1, S, and G2, same. And what that means is there is going to be a replication of the genetic material during the S phase. Okay, we're going to go from single-stranded to double-stranded chromosomes in interphase, in meiosis, just like we did in mitosis. Now, that sort of seems silly, okay? Why do that? Why double the genetic material if all we're going to do is try to cut it in half? Why don't we just cut it in half in the first place and be done? Well, again, because meiosis evolved after mitosis, and we're kind of stuck with this cellular machinery. Natural selection has evidently seen no real reason to change it, so it does things this way. We are going to, again, during interphase, during the S of interphase, we're going to turn these single-stranded chromosomes into double-stranded ones. And all of meiosis is going to be about then cutting this chromosome number in half. Okay? So the way we're going to do that is we're going to divide twice. Okay? So we have this DNA replication, because that's what we're stuck with, and then we're going to divide the cell once. We're going to separate the homologous pairs that we've seen, and then we're going to divide again and pull these chromatids apart. So here in the first division, we're going to separate homologous pairs from each other, and then in the second division, we're going to just pull the chromatids apart. Okay, so meiosis, two divisions. First, separate pairs. Second, separate the chromatids, like we do in mitosis. Okay, so let's have a quick overview of meiosis. Okay, what you see here is just the regular interphase, just like before in mitosis, but we are going to see a prophase that is going to look different, and we're going to talk about some of the differences in prophase, but this is one we need to watch because this is going to be different in meiosis, as will metaphase be different and anaphase here be different. Please notice that it's a 1 and a 1 and a 1, because obviously we're going to divide again, and we will call those divisions 2. Okay, now this here is telophase 1, all right? And basically from this point on, things are going to look a whole lot like mitosis. Things are going to look a whole lot like mitosis. It's not mitosis, but it is going to look like that, because from here on out we're going to separate chromatids just like we did in mitosis. So what we will see here is a repetition of the PMAT, prophase, metaphase, anaphase, telophase, but now our goal is just to separate chromatids instead of chromosomes. Okay? So please notice up here 2n. N is the haploid number of chromosomes, the haploid number. So in a human, N equals 23. 2n is our diploid number. Whoops, 2n is the diploid number, so that would be in a human 46. So here in this cell that's not a human, because it's only got 4 chromosomes, 2n, or diploid number, is 4. And you see that by the time we get here in telophase 1, because we separate the homologous pairs, we have essentially cut the chromosome number in half by the end of meiosis 1. We've gone down to 2 chromosomes. From 4 to 2, we've cut the chromosome number in half. The rest of this is just a matter of separating chromatids, and so we end up with a chromosome number of 2 in the end. Okay? So there are essentially two really important events of meiosis 1 that do not occur the same way in mitosis. The first event is crossing over, and this occurs in prophase 1. So prophase is a place, prophase 1 is a place we are going to be different from mitosis, okay? In meiosis, we are very different. We are going to allow chromosomes, homologous pairs that are similar but not identical, to do something called crossing over. In crossing over, your homologous pairs essentially will come together and intertwine. And when they do so, they will exchange bits of DNA. Perhaps a bit of this dad's chromosome in blue would pop off and cling to mom's, and mom's chromosome would cling to dad's, and they would swap bits of genetic material. Why? Because that makes the chromosomes different, and that means variety. This is another way to create variety, crossing over. It is also called synapsis. If anybody throws that obnoxious word at you, it just means this crossing over and creation of variety in the genetic material. The two chromosomes, 4 chromatids, when they are together and crossing over like that, it's called a tetrad. That's just the name of the unit while they're together. Synapsis, you can't read because it's all covered up. Now, the other event in meiosis 1 that is important is when we separate homologous pairs and not chromatids. Remember, at mitosis, essentially the chromosomes line up with their centromeres on the center line. That's not what happens in meiosis. Okay? At metaphase 1, at least, in the first phase of meiosis, the chromosomes will line up, and they will line up on either side. The homologous pairs will line up on either side of this imaginary center line. Notice the centromere is not on center, so that at anaphase we're going to separate homologous pairs, not chromatids. What that means, we'll create a cell here and we'll create a cell here. Notice 1, 2, 3, 4 chromosomes here. Notice when we divide the cell like this cell here will have 1, 2 chromosomes. This cell over here will have 1, 2 chromosomes. After we divide the cell here, we have essentially cut the chromosome number. We will cut the chromosome number in half at this point. Okay? So, yes, there we go. So this would be after anaphase has occurred and essentially the end of the first meiotic division. Now, these cells will then divide again, and we will then separate these chromatids from each other in the second meiotic division. Okay? All right. So, a little more detail about crossing over real quick. Okay? The first thing we do is actually crossing over means, first of all, the chromosomes are going to come together and they will cross over. Bits of their chromatids will cross over. The DNA will break and we will see a swap of material right there. You can see that. Okay? And it's going to refuse and we will have 2 chromosomes that are now different from the chromosomes we started with. They have swapped some genetic material. Okay? That is going to add to the variety in the genes. Okay? And we'll talk more about that in just, well, this is a good term. New combinations of traits will occur. If dad had brown eyes, mom had blue, and we swap, now the brown-eyed gene is on this chromosome and the blue-eyed gene is going to be on this chromosome. So we are switching up the combinations of traits. This is also called genetic recombination, and it's a good way to get more variety. Okay? Now, meiosis 2, basically, The main event is just separating the chromatids at that point. It is very much like mitosis, okay? So in meiosis 1, we have the crossing over, we have the lining up of homologous pairs. Notice no centromeres on the center line. We split them apart and separate homologous pairs. Okay, and now down here we're lining up with centromeres on our center line, and we're just going to split the chromatids, and that's the second phase of meiosis, which is very much like mitosis. Okay, now all of this, meiosis and sexual reproduction, leads to variation in three main ways. Okay, and these ways are all forms of genetic recombination, reshuffling genes. We're not creating new genes, but we are shuffling old genes in new combinations. We do that first by something called independent assortment of chromosomes. Okay? In other words, let's go back here. And okay, when the chromosomes line up in metaphase 1, and we have one member of each pair on either side of an imaginary center line. Let's say the blue comes from dad and the red from mom. So red from mom, blue from dad. They could just as easily have aligned with the red on this side, so we might have had moms both on the side, mom and mom over here, dad and dad over here, with the blue one on this side. They could just as easily have aligned like that, or they could have aligned with this red one over here and the blue one over here. It doesn't matter. They can align in any way. The mom's chromosome can be on either side, dad's can be on either side. It doesn't matter. Because of that, and because the same is true for this pair up here, we can get different combinations of traits just because these chromosomes are not glued to each other and they don't have to line up in this arrangement every time. When we are going through meiosis, these can align any way they want in different cells that are going through meiosis. Okay? That is called independent assortment. The alignment of the homologous chromosomes in metaphase 1 is random. They can go on either side of the center line that they wish. Okay? You can end up with a combination that looks like that, or that, or that, or that. And that puts different combinations of genes ultimately in different cells. Crossing over is another way we can recombine traits by swapping out some genetic material, mixing alleles, like so, like we see over here. Okay? And finally, random fertilization. Having no control over which sperm fertilizes which egg. The sperms are all different from each other. The eggs are all different from each other because of all this independent assortment and of all this crossing over, and we have no control over which is going to fertilize which. And we can get many new combinations of offspring created from different sperm fertilizing different eggs. Okay? All of this recombination gives us new combinations of traits and different individuals, variety among individuals. And this is fundamentally what drives evolution, variation. If this guy has the best camouflage, then he is going to outcompete all the other beetles of his species that don't have as good a camouflage as he does. Maybe this guy gets seen by the birds all the time and he gets gobbled up. His genes won't get passed on to the next generation, but his will. And so over time, maybe the beetle population becomes to look more like him, and these guys slowly get eliminated. This kind of variation helps to drive natural selection, drive evolution. Okay? So, here is a cell and an ovary. Here is a cell and a testis. They go through meiosis and create eggs and sperm. Those eggs and sperm fertilize, come together, and combine their genes to create a zygote with 46 chromosomes. Okay? Then that zygote reproduces by mitosis and creates more and more cells, each of them identical to the first, all of them with 46 chromosomes in them. Okay? So that's where mitosis would come in, and we would create a baby eventually. And that's called development. All right? Okay. Just an interesting little note here. How much variation do we get? Sperm plus egg, any two parents. There are 70 trillion possible combinations of genes for any two parents. That is an incredible amount of variety, and helps to explain why even with 8 billion people on the or 7 billion, whatever we have on the planet, we can still all be unique unless you're an identical twin. And even then, there's reasons why there might be even tiny differences between identical twins. Okay? So, sexual reproduction does a great job of making us different while keeping us similar. Okay? We can see familial resemblances in famous brothers here. But still differences, resemblances but differences, and this is how genetics works. We have things that are going to be the same, but also things that will be different. Okay? Just a few last notes. This is meiosis in a male, and it creates sperm. Again, we start out with a diploid cell, we go through meiosis 1, meiosis 2, this is meiosis, the two divisions, and we create sperm cells. This is called spermatogenesis. This is just sperm that occurs in a male. Meiosis that occurs in a male to create sperm. Okay? It is a continuous process. It goes on for the life of the male. Billions and billions and billions of sperm created over life. Okay? So, egg production or creation of eggs, meiosis in a female is called oogenesis, and it's a little bit different for one important reason, and that is that meiosis is not equal. In a female, we will create one cell that's got all the cytoplasm, all the organelles, all the nutrients, all the everything, and another cell that has almost none of that, and it just has some genetic material. So this has got the genes of the mother and lots of other stuff. This has the genes of the mother and almost nothing. And we will do that twice with our meiotic division. And the reason is we basically, in a female, the choice is made to have a cell that's got everything that baby could possibly need. It's a really awesome cell. And it is better to make one really awesome cell than 4 okay cells. So we basically are just getting rid of these and saying we're going to take that loss in favor of making one really great cell. Whereas if you go and look at male meiosis, you can see we end up from one diploid cell, we end up with 4 haploid cells, and they're all basically the same. Basically, a sperm is the male's genes. It's got some mitochondria, and it's got a tail. Mitochondria for energy, tail for swimming, genes because that's what we have to deliver to the egg, and that's it. Whereas the female is going to have everything that baby needs in this cell, and plus her DNA, and is going to be much more providing much more for the baby than the male contribution. Okay? We're just not going to worry about all this stuff because some of it's not true anymore. Alright, you should recognize what a polar body is. This is that little rejected piece of female DNA, the little non-developed egg, if you will, that's a polar body. It's an unequal division. And let's see. Just be aware that meiosis happens a little differently across kingdoms. Here is the classic animal plan where you are haploid only in the gamete stage. You spend your entire life as a diploid organism and create gametes for just a brief time that will get fertilized and will become diploid again. Okay? Whereas certain algae and fungus spend most of their life actually being haploid, and only a very short part of their life being diploid. Fungus are great at this, and if you'll look, there are certain plants like ferns and whatnot that will spend about half and half, haploid versus diploid. So there's different kinds of patterns involved. Okay. And there we are. We're very much like that frog. Okay, that's it. And I will see you next time when we're going to talk about what Mendel did and all those little Punnett squares that I know you love so much. Take care. See you later.